Meiosis, which is the second topic which forms part of paper 2, can be seen as one of the easiest topics that can be used to score marks in your NSC final exam. Therefore, in this video, we will be using questions from section B to revise this topic to see how you could possibly score marks very easily in meiosis. Some of the key points that you could focus on when studying this topic are firstly identifying the main characteristics of each phase, explaining crossing over, where you have homologous chromosomes that form bivalents and not chromosomes only. Using the process with a diagram and label, so you need to understand the process using a diagram and labeling the diagram. The significance of meiosis, why is meiosis important? For the production of haploid gametes, the halving effect of meiosis overcomes the doubling effect of fertilization, thus maintaining a constant chromosome number from one generation to the next. The mechanism to introduce genetic variation through crossing over and random arrangement of chromosomes on the equator. This is extremely important. You may be asked to describe how meiosis introduces genetic variation. There are two processes that occur, namely crossing over occurs in prophase 1 and random arrangement of chromosomes on the equator occur both in metaphase 1 as well as metaphase 2. Then identify the main characteristics of each phase. So these two major forms of meiosis. You have meiosis 1 and meiosis 2. You have prophase, metaphase, anaphase, and telophase in each of meiosis 1 as well as meiosis 2. Then you need to explain crossing over, where homologous chromosomes form bivalent chromosomes, not just chromosomes. The process of crossing over using diagrams with labels. The differences between meiosis 1 and meiosis 2. The similarities between mitosis and meiosis, especially in different phases. For example, in metaphase 1, chromosome pairs align, and in metaphase 2, single chromosomes align. Abnormal meiosis, you need to understand nondisjunction and Down syndrome. Nondisjunction of chromosomes occur at position 21 during anaphase in humans to form abnormal gametes with an extra copy of chromosome 21. Please make sure you are able to understand the key focus points which have been highlighted in this particular slide. The first question that we'll look at is taken from question 2.5 that reads, the diagram below represents two phases of meiosis. So you have diagram A as well as diagram B. You can see in diagram A you have chromosomes arranged on the equator. In diagram B, you also have chromosomes arranged on the equator. However, these chromosomes are still going to be coming in the homologous chromosome pairs. 2.5.1 asks you to identify the phase represented by diagram B. So if we have a look at diagram B, we are trying to identify this phase. Because we still have our homologous chromosome pairs, therefore we are saying it's metaphase 1. 2.5.2 says describe the events in the phase that follows diagram A for 3 marks. You are describing the events in the phase that follows diagram A. You are not simply describing the events in this phase. You are describing the events in the phase that follows diagram A. If diagram A is going to be metaphase 2, why metaphase 2? Because we have individual chromosomes on the equator, then it follows that the next phase will be anaphase 2. So we are simply describing anaphase 2. So for 2.5.2, your spindle fibers are going to shorten, your centromeres are going to split, separating each chromosome into 2. Daughter chromosomes are pulled to opposite poles. So you'll have chromatids moving to the opposite poles in anaphase 2. 3 points for 3 marks, question 2.5.2. 2.5.3 says name one place in the human female where meiosis takes place for 1 mark. One place in the human female. In females, meiosis is the process of gamete formation. And gamete formation, gametogenesis in females generally takes place inside the ovaries. Therefore, the answer is the ovaries. The next question, 2.2, says the diagram below shows different phases of meiosis in an organism. So you have diagram 1, diagram 2, and diagram 3, each showing you phases of meiosis. The first question, 2.2.1, says identify structures A for 1 mark. So if you look at structure A, it's pointing to the nuclear membrane. Structure C is pointing to the spindle fibers. Then 2.2.3 says give two observable reasons why chromosomes B above are regarded as homologous. For 2 marks, so chromosomes B above, so we are looking at these chromosomes, why are they referred to as homologous chromosomes? Based on observable reasons, we can see the chromosomes have the same size, they have the same shape, and they have the same centromere position. And those are general characteristics of homologous chromosomes. Therefore, we can say they have the same size or length, they have the same shape, and they have the same centromere position. Any two of those observable reasons would have given you 2 marks for 2.2.3. Then the next question, 2.2.4 says identify the phase in diagram 1. So we are identifying this phase in diagram 1. Without any doubt, it's going to be prophase 1. Question 2.2.5 says during the phase identified in 2.2.4, a process takes place that leads to genetic variation. Name and describe the process for 5 marks. So during the phase identified in 2.2.4, what was that phase? Prophase 1. A process takes place that leads to genetic variation. Name and describe the process. So the process that occurs in prophase 1 that leads to genetic variation is going to be crossing over. Therefore, we are going to first name crossing over. You get 1 mark for naming crossing over. Then you describe crossing over. Firstly, crossing over occurs during prophase 1. Homologous chromosomes or non-sister chromatids are going to overlap and they're going to meet at a point where they touch called chiasma, which is mater. Genetic material is going to be exchanged, resulting in new combinations of genetic material. The next question, 2.2, says the diagrams below show the same cell at different phases of meiosis. The same cell at different phases of meiosis. So you have diagram 1 as well as diagram 2, but they are showing different phases of meiosis. The first question, 2.2.1, says identify part B. So if we're looking at part B, it's pointing to this dotted shape structure, referring to the spindle fiber. Then the phase of meiosis shown in diagram 2, we are looking at the phase that occurs here. We can see that crossing over is going to occur. Therefore, without any doubt, it's going to be prophase 1. State one function of part A. So if we look at part A, it's pointing to that dotted structure that's going to hold the spindle fibers, referring to one function, because they're saying give the function, so it's going to be the centriole, and therefore the centriole is going to form the spindle fibers. Then one function of part C. If we go to part C, that's referring to the centriole. Part C refers to the centromere. And what does the centromere do? It's going to hold your chromatids in place. It's going to join your 2 chromatids together. The next question, 2.2.3, says tabulate 2 differences between the phase shown in diagram 1 and the same phase in meiosis 2 for 5 marks. 2 differences between the phase shown in diagram 1. So what is the phase shown in diagram 1? Here we have our chromosomes moving towards opposite poles. And the same phase in meiosis 2. So this is going to be anaphase 1. But we're giving the differences between anaphase 1 and anaphase 2, because anaphase 2 forms part of meiosis 2. Therefore, we're going to draw the table and we're going to say anaphase 1 as well as anaphase 2. In anaphase 1, no centromere is going to split. You see the centromeres are still intact. But in anaphase 2, the centromeres are going to be splitting. In anaphase 1, your chromosomes move to the opposite poles. You see the full chromosomes are moving to the opposite poles. But in anaphase 2, your chromatids move to the opposite poles. In anaphase 1, a homologous pair of chromosomes moves to one pole as abnormal process D. So you see here, a homologous chromosome pair is pulling the entire pair to the opposite poles because of the abnormal process. But if it was anaphase 2, a whole chromosome would move to one pole as abnormal process D. You wouldn't have had a homologous chromosome pair. It would simply just be one chromosome moving to the same pole. The next question, 2.2.4, says explain the effect of abnormal process indicated by D if it occurred in chromosome pair number 21 in humans for 4 marks. What are we doing here? We are simply explaining what happens in Down syndrome. So firstly, 2.2.4, one gamete will have an extra chromosome, or it will have 24 chromosomes. And when it fertilizes a normal gamete with 23 chromosomes, then the zygote will have 3 copies of chromosome number 21, or you can say the zygote will have 47 chromosomes. This results in Down syndrome. So one gamete will have an extra chromosome, which has 24 chromosomes. If that chromosome fertilizes with a normal gamete that has 23 chromosomes, then the zygote will have 47 chromosomes instead of 46, and this results in Down syndrome. The next question, 2.3, says the diagram below represents metaphase 1 of meiosis. Metaphase 1 of meiosis. 2.3.1, identify part B. If we look at part B, it's the structure on the outside. That part is going to be referred to as the cell membrane. 2.3.2, state one function of part A. Part A is pointing to the dotted shaped structure on the outside that's holding the spindle fibers in place. So therefore the function would simply be it produces the spindle fibers and it holds the spindle fibers in place. The next question, 2.3.3, says explain the contribution of metaphase 1 to natural selection for 6 marks. So metaphase 1, we know that in metaphase 1, something that contributes towards increasing genetic variation is going to be random arrangement of chromosomes on the equator. If we increasing genetic variation, we know that if we're speaking about natural selection comes from the topic on evolution. And to start explaining our process of natural selection, we normally say that we have a great deal of variation that occurs in a population. You are simply speaking about the events of metaphase 1 that increases genetic variation, and then you are using that to describe the process of natural selection, and that is how you are awarded 6 marks. So how do we start? Firstly, random arrangement of chromosomes occur at the equator, resulting in the formation of genetically different gametes. This leads to increased genetic variation in a population. That coincides with the first sentence that we normally have in describing natural selection. Therefore, we simply go on now from natural selection. This will cause some individuals to have favorable characteristics and some will have unfavorable characteristics. When the environmental changes or if there's competition, then those with the favorable characteristics will survive. Those with unfavorable characteristics will die out. And that is how you get 6 marks for 2.3.3. The next question says the diagram below shows 4 possible daughter cells formed at the end of this meiosis. So for 2.3.4, you're going to draw cell D including the missing 4th chromosome. So cell D including the missing 4th chromosome. If you look at the chromosomes here, you have a white, and a black. You see that B and C, there's similarities between B and C because all the chromosomes are going to have half black and half white. But in A and D, it's a full white and 3 full blacks. Therefore, in D, you're supposed to have 3 full whites and 1 full black. So you're simply going to draw that structure because you're including the missing 4th chromosome. That missing 4th chromosome would be that last white one that still needs to come in there. Therefore, the answer looks like this. You have your last white chromatid that was left open. You included it. It was not shaded. Therefore, 1 mark for the cell D copied correctly. The missing chromosome has a straight shape and the missing chromosome is unshaded. 3 marks for 2.3.4. The next question, 2.2, says read the information below. Down syndrome occurs in people of all races and economic levels. It occurs as a result of mutation during meiosis. Individuals with Down syndrome suffer from learning delays, distinct physical features, and poor muscle development. The maternal age has an impact on the risk of Down syndrome. The table below shows the relationship between the maternal age and risk of Down syndrome. So you have your maternal age 25, 31, 35 and 40, and you have your Down syndrome risk 1 into 1 in 1250, 1 in 1000, 1 in 400 and 1 in 100. 2.2.1 says state 2 symptoms of Down syndrome from the information above. So you have the symptoms listed here. You can simply say 2.2.1, learning delays, distinct physical features, and poor muscle development. Then 2.2.2 says identify the maternal age. Is it 25, 31, 35, or 40, which is expected to have the greatest risk of having a child with Down syndrome for 1 mark. So we look at the risk. We look at the highest possible risk, which is 1 in 100, and the age is going to be 40. Therefore, the answer is 40. 2.2.3, you're calculating how many times did the risk of Down syndrome in females increase between the age 35 and 40 and show all your working. So you go to 35 and 40, you see it's either 1 in 400 or it's 1 in 100. But how many times? So you want to change the unit of measurement, therefore you are going to divide. So 400 dividing by 100 is going to give you 4 times. 2.2.4, you are describing the events of the phase of meiosis 2 that results in an abnormal gamete that leads to Down syndrome for 2 marks. What is going to occur in meiosis 2 that's going to cause an abnormal gamete that leads to Down syndrome? In meiosis 2, in anaphase 2, chromatids at position 21 fail to separate, or non-disjunction of chromatids occur, leading to an extra chromosome at pair number 21 in the gametes. That brings us to the end of this video where we revise the topic of meiosis using section B questions. Please ensure that when you are studying for paper 2, you are studying DNA, you are studying meiosis, you are studying genetic and inheritance as well as evolution. The next video will use genetic and inheritance using section B questions and revise this topic. Thank you very much for taking the time to watch this video. I hope to have you all join me in the next video, which will be based on genetics and inheritance using section B questions taken from previous exam papers.